Hello everyone, today we are going to talk about embryology. We are going to discuss the structure of the spermatozoon, oocyte, how they fuse one with another, so the fertilization process, the cleavage, and up to the process of implantation. This topic is very interesting because nowadays assisted reproductive technologies are very widespread because lots of couples, they suffer from infertility problems, and there are many ways how to solve this issue. And another reason why embryology is so relevant nowadays is that the regenerative medicine is developing very actively, and to understand the potential of every cell in the adult organism and to understand how we can retransform one type of cell into another, or what stem cells to use to obtain the tissue we need to understand perfectly the normal process of their development along the embryogenesis. So today lots of interesting aspects are going to be discussed. Let's start from the structure of the spermatozoon. First, this is the cell with haploid number of chromosomes destined to fertilize an egg. So the nucleus is very small, and this is the smallest nucleus in the human body, as it contains 23 chromosomes which are tightly packed. They are not used. There is no euchromatin in this nucleus, and as you can see by the shape, the nucleus is flattened. This is the top view and this is the side view. Okay, so the head is tapered a little bit and its end to facilitate the movement. So in the structure of the spermatozoon, everything's fit for a long journey, as the fertilization takes place only in the upper 1/3 of fallopian tube. So spermatozoon need to travel for a relatively large distance, and only those with a perfect shape, with enough energy, will reach their purpose, the objective. Okay, let's consider it in details. The head of spermatozoon, it contains, except the nucleus, the acrosome. Acrosome, this is the modification of the Golgi complex. It is sometimes called like a cap, and it contains a set of enzymes, digestive enzymes, helping spermatozoa to penetrate the zona pellucida. We'll discuss a little bit later. So to fertilize an egg, spermatozoa need to cross a barrier, and exactly these enzymes help spermatozoon to do that. Then except the acrosome and the nucleus, in the head there is the plasmalemma covering it, and after that there is a neck region. In the neck region, centriole is located. Then the very critical part of the spermatozoon is the tail. It has 3 parts, and now you have to be attentive to not to confuse them. So middle piece is the first part, because it is not in the middle part of the tail, it is in the middle part of the spermatozoon. So middle piece is the first. Then principal piece, it is called so because it is the longest one, and end piece, the short end piece. Then let's investigate them in details. So the middle piece, it contains the so-called exonemma. Axis is the axis, nemma means a thread, so the axial thread is located in the center. It consists of 9 pairs of microtubules and a pair inside. So we describe the structure of the exonemma by the formula 9 into 2 plus 2 means 9 pairs in the perimeter and a pair in the center. Then exonemma is surrounded by fibrous dense fibers. There are 9 dense fibers, and they increase the thickness and the beating force of the tail. Besides, the movement of the tail is due to the sliding of microtubules in the exonemma, one along another, and it uses the ATP energy. To get ATP, it needs a mitochondria. And there are a lot of mitochondria in the tail, exactly in the middle piece. So middle piece contains those 3 elements: the exonemma, then those coarse fibers, so dense fibers, and mitochondria around them. Then principal piece. Principal piece, it has almost the same structure except mitochondria. Instead of mitochondria, there is a fibrous sheet in the middle, in the principal piece. The fibrous sheet consists of ribs, one and the second, and together they restrict the movement of the tail in the horizontal plane. That's why the beaten tail of the spermatozoon is not spiral in three-dimensional space, but it is oriented in the same plane in a whiplash-like manner. Let's look at this video. So as you can see here, this is a very slow one, but here the normal spermatozoon, and you can see the manner in which the tail beats. Out of that, the end piece contains only axonema, so fibrous ribs and coarse fibers, they are absent there. So the end piece is the most thin. And if we look at the tail with the transmission electron microscopy, we can distinguish here all three parts. For example, here we can see the middle piece, as we can find here axonema in the center, then those coarse fibers, so dense fibers, and around them there is a huge mitochondrium. So this is the middle piece and the plasma membrane around it. Then here we can't see mitochondria, but axonema and fibrous sheet. So this is the principal piece, and this is the end piece, because only axonema is surrounded by the plasma membrane. Very easy to distinguish. Yeah, and also we have to mention that in the head of the spermatozoon, I forgot to say that in the nucleus, the hypercondensation of DNA is acquired due to special proteins called protamines. They are used instead of histone proteins. And instead of 30 nanometer fibril, which is formed out of nucleosomes in the conventional chromatin, special structures called toroids are formed. So in this way, DNA is organized more compact, comparing with the nucleus of the interface cell. Okay, so after we have considered the spermatozoon, its head, its tail, three parts of the tail, let's talk about the problems, because why we investigate the normal structure, because sometimes spermatozoa could be defective. For example, like in this case, what will be shown. For example, you can say the bent head. They have sometimes double head or double tail. Some of them are very slow guys, as in this case. You can compare its slow motion due to probably too long tail. You can compare its movement with others. So defective spermatozoa could be present, and sometimes it causes the infertility problem. And semen analysis is the first procedure which is prescribed in this case. What is analyzed and what could be revealed in this case? So you have to know some terms associated with this analysis. Sometimes there is no ejaculate at all. Sometimes there are no spermatozoon in the ejaculate, so we call this azoospermia. Then maybe there is only a few spermatozoa there. Maybe there are a lot of spermatozoa, but they are immotile, or there are many abnormal spermatozoa in the sperm. So all those phenomena could be the reason of infertility, and that's what could be revealed with the semen analysis. Also, the sperm concentration, their total quantity, is assessed. And the motility. So as for the quantity, the more the better, as it is written, but we have to comprehend that to fertilize an egg, there should be millions of spermatozoa, hundreds of millions in the seminal fluid, and 15 millions per millimeter. So at least several 15 to 12 to 20 millions, and only several hundreds would reach the oocyte. And it is important because only one spermatozoon can't fertilize an egg. Notice that up to 50% of spermatozoa could be defective. That's because along their journey, along this competition, lots of them die, and this is the normal process. So abnormal spermatozoa, they will undergo negative selection, and they will have no chance, because all types of abnormalities, they result in the bed motion. So if they have any defects, they will swim much slower than their rivals, the competitors. In this way, evolution get rid of defective ones. Okay, let's move on. And as today we are going to talk about assisted reproductive technologies, we have to mention the intracytoplasmic sperm injection. This procedure is used exactly in case when inside the ejaculate there are only a few spermatozoa, or they are immotile, or there are a lot of abnormal spermatozoa there. So in these cases, spermatozoon is chosen under the microscope, or by the specialist. So many parameters are assessed. First of all, motility and morphology. That's why we investigate morphology in such details. And after that, this spermatozoon is aspirated in a microneedle, and that's what happens. Micropipette holds the oocyte surrounded by the zona pellucida, and microneedle injects. You see, the procedure is rather harmful, and the whole spermatozoon now would be injected in the cytoplasm. Notice that all parts of spermatozoon appear inside the oocyte. So the tail with its plasma membrane and everything will be digested by the oocyte's enzyme except the nucleus, except the centriole. All other structures, including mitochondria, will degenerate. Okay. So intracytoplasmic sperm injection helps to obtain embryos even in case of severe male infertility problems. You know that nowadays, even in case of total absence of spermatozoa, there are some approaches helping to obtain spermatozoa out of stem cells. So just regular stem cell could be used to produce spermatozoa, and further they can be used to fertilize the oocyte. Okay, then, as we have discussed the structure of spermatozoon and different problems associated with that, we have to discuss the structure of the oocyte. So this is the female gamete cell with 23 chromosomes in it, and it differs from the spermatozoon as it is huge, comparing with the sperm, 170 micrometers in diameter. That's because it contains all the nutrients necessary for the first week of development, as spermatozoon it carries on the nucleus and the centriole. Then oocyte is immotile, so it can't move itself, it can't move and it is moved passively by the beaten cilia of the fallopian tube. Then first we have to discuss the envelopes it has. These envelopes, they represent obstacles on the way of the spermatozoon, so we have to know. The first one, corona radiata, or the layer of follicular cells. These are the supporting cells, like servants. They provide the oocyte with nutrients and takes back wastes. So they help oocyte to survive out of the ovary. Then zona pellucida. This is the shining zone made up of glycoproteins, among which ZP3 is the most important. They are called ZP because it's zona pellucida. This is the abbreviation. ZP3, it functions as a ligand for spermatozoa receptors. Exactly this glycoprotein will be recognized by the spermatozoon. So spermatozoon could attach to the zona pellucida only in case it has this glycoproteins. Then plasma membrane or the oolemma. This is the last obstacle, and this is the last part, the last border, like that. So 3 envelopes oocyte has, and what's there inside its cytoplasm? It's very interesting to comprehend that it has no nucleus, because the oocyte, just after the ovulation, is in the metaphase of the second meiotic division. So only in case it is fertilized, the resumption of this division takes place. In case oocyte is not fertilized, it degenerates, and this division never ends. So there is no nucleus, but only metaphase plate. Then, also it contains cortical granules. They are not shown in the diagram, but they are called cortical because just under the olema, just under the plasma membrane, in the cortical superficial layer of the cytoplasm. And as for their function, we will discuss later. And of course, your granules, they are dispersed uniformly in the cytoplasm, and we have to understand that in humans, nutrients along the pregnancy are obtained from the maternal blood. So yolk is needed to survive only during the first week of development. That's why human oocytes are oligolecytal. You have to know this term and what does it mean. Oligo means a little, lecytal means yolk. So oligolecytal means a little yolk. We don't need too much, yeah, as birds, for example. Birds, they use the yolk along all the embryonic period. So, of course, their oocytes are polylecytal. Then, due to distribution of the yolk, we call our oocytes isolecytal. Iso means equal. Isolecytal means that yolk is equally distributed in the cytoplasm. So, in case of birds, for example, they have telolecytal oocytes or centrolecytal oocytes when yolk is concentrated in the center or at only one pole of the oocyte. In case of humans, we have equal distribution, so we call oocyte isolecytal. Okay, let's move on. Now, as we have discussed the structure of the spermatozoon and the oocyte, let's talk how they fuse and what are the phases of their interaction. Fertilization could be subdivided into 3 steps. The first is the distant interaction of gametes. So when spermatozoa appear in the female reproductive tract, they start to interact with oocyte, even despite they are not in contact, but they feel chemicals and they decide where to move. They choose the direction of the further movement. So we call this distant interaction of gametes. Then contact interaction. When they come in contact, they realize several programs and several reactions we are going to discuss later, and they have the very high biological significance. And after that, fusion, when cells, they fuse one with another. So let's discuss in details every out of those 3 steps. The first step is the distant interaction. It includes the positive chemotaxis. What it means? Chemotaxis means that chemicals attract the spermatozoa. Positive means that they attract. If, for example, negative means that they deter. Yeah, but in case of spermatozoa and oocyte, oocytes produce chemicals attracting spermatozoa. And this phenomenon is called positive chemotaxis. Something like spermatozoa smell the oocyte and they move towards the smell. So they choose where to go. Then next phenomenon is negative rheotaxis. Rheos means the current of the fluid, and negative means that when spermatozoa detect the movement of the fluid, they swim against this current. So they have mechanism activating them in case they move against the current. It's very important because in the female reproductive tract, fluid moves from fallopian tube to the uterus to move the newly fertilized organism, newly created organism, or the oocyte from fallopian tube, from the ovary to the uterus, and there is the natural movement of the fluid. But spermatozoa need to go in opposite direction from the uterus to the fallopian tube to fertilize an egg. So they move against the current of the fluid. That's what we call negative rheotaxis. And the last reaction, which is very important, is called capacitation. Capacitation is when spermatozoa become capable to fertilize an egg. It is very important because if we take fresh spermatozoa from ejaculate and mix them with oocytes, they are not capable to fertilize them, because to become capable, For fertilization, spermatozoa need to be incubated in the female reproductive tract at least for 7 hours. So if less than 7 hours, they can't perform their function. Capacitation is when spermatozoa swimming against the fluid current, and at the same time they are washed. So it is compared with the washing of the head, because the head need to be clean enough to interact with the receptors in the zona pellucida. Because in the seminal fluid there are lots of wastes, lots of rubbish material, and lots of glycoproteins, and they help to protect acrosome from preliminary acrosomal reaction. They protect the acrosome. But when spermatozoa are already in the female fallopian tube, this protection should be removed, and that's what we call capacitation. You know that some contraceptives they are used to release acrosomal enzymes preliminary, and in this case spermatozoa, when they come to the fallopian tube, even in case they reach the oocyte, they can't fertilize as they have no enzymes. Okay, and in this funny video file, the comparison is shown between the spermatozoa and the human and the humans to reveal how difficult for spermatozoa to realize their function, because once they are released in the female reproductive tract, there are millions of them, and tens of millions, and they have to climb up to swim against the current of the fluid, and they compete with each other, and lots of them, predominant amount, is going to die because of the competition, because of the harsh environment, and also because they fight against the immune system of the female organism, as for female spermatozoa are foreigners. So in this competition, only the most strong and healthy, the most proper, survive and reach their objective. So when they come in contact with the zona pellucida, they start to attack it, and they can fertilize it only in case there are several hundreds of them. Okay, let's move on. So in details about the capacitation. Capacitation, it includes the increased level of cyclic AMP. Then also the phosphorylation takes place, tyrosine phosphorylation, which is used as the biochemical marker of capacitation. That's why you have to know about that, because for in vitro fertilization the capacitation should be assessed to use spermatozoa properly. Then also increased calcium concentration takes place in the spermatozoa tail, and as I told you before, glycoproteins, different molecules are removed from the head, and also the cholesterol is also removed, making membrane more soft and prepared for the acrosomal reaction. So capacitation is a complicated process, but it is very important, and it lasts nearly 5 to 7 hours in the fallopian tube. Then, as a result of capacitation, spermatozoa, they become more active, and that's the phenomenon we call hyperactivation. So just after the capacitation, the hyperactivation of spermatozoa takes place. The beating of the tail becomes more forceful, and that's due to the increase of calcium inside the spermatozoon. Calcium channels, to increase the intracellular concentration, calcium channels are located in the tail of the spermatozoon, in the membrane exactly of the tail, and those proteins are called CADSPERs, cationic channels of the spermatozoon. So the channels for the calcium. Mutations in those proteins result in infertility problems because spermatozoa are not active enough to reach the oocyte. Then different factors activate spermatozoa, as we have already discussed: calcium, cyclic AMP, proper pH level, and those changes are regulated by the proper phosphorylation. And that's why modulation of some enzymes participating in phosphorylation process is used in practice to stimulate spermatozoa motility. For example, the stimulation of protein kinase A or inhibition of protein phosphatase are used to improve the activity of spermatozoa, to overcome the infertility caused by the low sperm motility. Okay, so after we've discussed the distant interaction between the gamete cells, when spermatozoa feel the smell of the oocyte, they move against the current of the fluid, and along with this movement and swimming, they are washed and become hyperactive due to calcium and cyclic AMP. Now we approach to the contact interaction of gametes. So when spermatozoon comes in contact with the oocyte, it interacts, it starts to digest the zona pellucidum, so enzymes are released out of the acrosome, and in this process the membrane of the acrosome is fused with the membrane, plasma membrane of the spermatozoon, and enzymes are expelled. They start to digest the zona pellucida, but what's interesting is that the enzymes of only one spermatozoon is not enough, the amount, and only several hundreds need to be present around the oocyte to properly fertilize it. And this phenomenon has high evolutionary significance, because in case there are only a few spermatozoa, they could be defective, so they can't participate in the competition. Only several hundreds are needed. That's why if there are a few spermatozoa in the ejaculate, or lower number of spermatozoa, or increased percentage of abnormal spermatozoa, it results in infertility problem. So acrosome reaction is realized, zona pellucida is digested, and after that the receptors on the head of spermatozoon, they come in contact with ZP3 glycoproteins in the oocyte, and it has a very high significance, because in this way the interspecies breeding is prohibited, because spermatozoa of only one biological species so can fertilize the same oocyte, and breeding between different species is impossible because of the specificity of the receptors on the head of spermatozoon and ligands in the zona pellucida of the oocyte. Okay, so acrosomal reaction is when acrosome releases its enzymes, and as I told you, if acrosome preliminary loses its enzymes, then such spermatozoa are infertile. Then, as for the oocyte, after spermatozoa have released their enzymes to digest zona pellucida, when the only one and the first spermatozoon has entered the cytoplasm of the oocyte, so just after the only one and the first has penetrated the ovulema, oocyte realizes its reaction. The first one is depolarization of the ovulema, so it is called fast block to polyspermy. Just after one has entered, depolarization deters other spermatozoa. Second is cortical reaction. Due to depolarization in the oocyte, calcium increases in its concentration, and it results in the exocytosis of cortical granules. So just under the cytoplasm there are lots of cortical granules, and they are released to the perivitelline space, and those enzymes, they stiffen the zona pellucida, and this is what we call the zona reaction. When zona pellucida becomes from jelly-like, it becomes more stiff and indigestible, impenetratable for other spermatozoa. So together, taken together, those three reactions, they help oocyte to protect itself from more than one spermatozoon, because to restore the normal number of chromosomes, only one spermatozoon is needed, because humans, we have stable number 46 chromosomes, and zygote should have 46. In case we have more numbers, such organism, polyploid organism, can't develop. So if oocyte will be fertilized by more than one sperm, it will be spoiled. So to prevent that, those reactions are needed. So first depolarization, then increased calcium results in the exocytosis of cortical granules, and zona pellucida becomes stiff and impermeable for others. And now it is called fertilization membrane or perivitelline barrier. Let's watch this video, very helpful. Here the oocyte, human oocyte, is shown under the microscope, and now spermatozoa are added to the solution, and you can see those small cells, these are spermatozoa exactly, attacking the oocyte, trying to fertilize it. This procedure is used in vitro fertilization. In case fallopian tubes are blocked, for example, in case natural fertilization is impossible, because spermatozoa, they can't find an oocyte, the oocyte, because the fallopian tubes are obliterated due to infections, due to surgical interventions, so many other reasons. So this is the very distributed, frequently occurring reason of female infertility. So spermatozoa obtained, they are incubated, capacitated, and added to the oocytes. Now you can see here the fertilization membrane appears. It means that one spermatozoon is already inside, and as a result this membrane was formed. So this is the confirmation of the successful fertilization process, and as you can see, usually several oocytes are obtained for in vitro fertilization because embryos survival is not very high. That's why several embryos are usually created, and several embryos are used to implant to the uterus, to increase the chances of the pregnancy. Okay, let's move on, and now we talk about the fusion of gamete cells. So the third phase of fertilization. You remember we have discussed the distant interaction, then contact interaction. When they come in contact, they release enzymes to treat each other. And after that, they at last fuse. What happens inside the fused gametes? First of all, spermatozoon brings its activating factor, stimulating oocyte to divide, and this is enigmatic factor. It was revealed that phospholipase CZ1 is one which is needed to activate an egg, but there is also another factor, it is unknown up to now, which stimulates oocyte to divide. Then what happens? The female metaphase plate resumes the second meiosis, and female prenucleus is formed. As I told you, the oocyte just after the ovulation, it has no its own nucleus, it is in the metaphase. So only in case spermatozoon fertilizes this egg, metaphase plate then goes to anaphase, and the second meiosis is complete only in case of fertilization. So female prenucleus is formed and the second polar body. Then male DNA, as I told you, male nucleus is hypercondensed and it doesn't use its own genetic information. So coming to the oocyte, it replaces all its proteins with the histone proteins and form conventional chromosomes. Then male prenucleus is formed, and as you can see, this is the stage of two prenuclei. Here two prenuclei are present inside the same cell, so now this is the zygote, but two nuclei not yet fused one with another. So the next step is the synthetic period. Synthetic period when DNA replicates. DNA is replicated and sister chromatids appear in both nuclei, in both prenuclei. So as we remember, oocyte has 23 chromatids and 23 chromatids in the spermatozoon. But to undergo the first division, the number of chromatids should be duplicated, because daughter cells also should have 23 paternal, 23 maternal chromosomes. So synthetic period is needed. And only after that, the first division takes place. And let's summarize all those processes in this video. The full link is in the description below. The video is very helpful, so let's watch. So spermatozoa are shown in the female reproductive tract. As you can see, there is a harsh environment and lots of spermatozoa die, and many of them enters through the cervix. They enter the uterus during the intercourse. Then inside the uterus, as we can see, lots of corpses, as lots of spermatozoa are defective, and it's a normal process, and some of them engulfed by the female immune system. As we can see here, it's the leukocyte, and it digests phagocytous spermatozoa. So some spermatozoa are the victims of the immune system. Then two fallopian tubes. Here the uterus is shown, and as you know, every menstrual cycle only one oocyte is released, only in one of those two ovaries, and oocyte produces chemicals attracting spermatozoa, as we have discussed, the positive chemotaxis. So the chemicals in this video are shown with the help of light. So enlightened fallopian tube means that oocyte is exactly there, and spermatozoa they move in this direction. Here the oocyte, surrounded by follicular cells, is moved by the beating of the cilium. Spermatozoa are moving in opposite direction, and this is what we call the negative rheotaxis. Negative rheotaxis, when it becomes active, when it moves against the current of the fluid, it gets washed, and that's what we call capacitation. So they become hyperactive now, because capacitation was performed, and only capacitated and clean spermatozoa, moving against the current of the fluid, they can squeeze between follicular cells. So they do not digest follicular cells because there are wide clefts between them. At last, one of them has reached the zona pellucida, and now acrosomal reaction is realized. You can see the fusion between the plasma membrane of the spermatozoon and its acrosome. Acrosomal enzymes digest jelly-like zona pellucida, helping spermatozoa to come through the zona pellucida to the perivitelline space. This is the space between the ovolemma and the zona pellucida. So here is the winner, the only one in the first, and it comes in contact with the oocyte, and their plasma membranes fuse one with another with the help of integrins. You can see the fusion of the spermatozoa membrane and the oocyte. The tail is left behind. So, as the only one, spermatozoon is already inside, others should be prevented from entering, and here exactly the turquoise color granules are released, and this is what we call the cortical reaction. Cortical reaction stimulates the zona reaction. As you can see, zona pellucida is no longer jelly-like. Now it is stiff and impermeable for others, because ZP3 glycoproteins are degenerated, so spermatozoa can't come in contact. That's what's happening inside the cell. We can see uncoiling of paternal DNA and formation of conventional chromosomes with histone proteins. So male pronucleus is formed in this way with only one chromatid in each chromosome, as you can see on the 23. Then resumption of the second meiosis takes place in the oocyte. So oocyte now also has formed its pronucleus with 23 chromatids. Now synthetic period takes place when they duplicate their chromatids. They fuse one with another, losing nuclear envelopes. They become organized in the same spindle, which is organized between the maternal and paternal centrioles. So now every chromosome should be divided into two sister chromatids, and the first division of cleavage takes place. Along with the cleavage, the zygote, a newly formed embryo, is passively moved by the beaten cilia of the fallopian tube to the uterus, and this process takes nearly 5 days. So this traveling is rather a long journey, And before this embryo reaches the uterus, the pregnancy can't be detected, so we do not talk about the pregnancy. That's only the result of fertilization. And lots of contraceptive drugs, they do not prevent fertilization, but they prevent the process of implantation. Okay, so let's discuss what happens further. Just after the fusion of gametes, after the formation of the zygote, cleavage takes place. So zygote starts to divide. And along with this division, there is no phase of growth, there is no G1 period, so cells do not increase in size because the amount of nutrients is restricted. Only oocyte has some yolk granules, and during the first 5 days, no external sources of nutrients are used, so only internal sources. That's why every daughter cell just after division is 2 times smaller than the maternal cell. That's why we call this cleavage, but not mitosis. We call cleavage because along with this process, cell do not grow, and each next division results in the formation of smaller cells. For human zygote, you should know the different characteristics of cleavage. First of all, cleavage in humans is complete, means that whole oocyte is divided, whole zygote is divided, then symmetric, means that daughter cells are equal in size, and asynchronous. Asynchronous means that blastomers, they do not divide together in the same time point, but instead they divide one by one. Let's look at this video. Let's watch this video. And here notice that this is the zygote and this is the polar body. There could be several polar bodies even. Polar body is the small cell which is formed as a result of meiosis. It contains only maternal chromosomes, but it doesn't participate in the development, okay? And the zona pellucida now, which is called the fertilization membrane, it surrounds the embryo up to the moment of implantation. So let's look how the cleavage is realized. So we can see the stage of 2 blastomers. After that, their number increases, and as you can see, they divide one by one. First, second, now this one has divided, so another one. As a result, you can see cells, they divide one by one, so the division is asynchronous, complete because the whole cell is divided, and symmetric cells are equal in size. Then also you have to know several other terms. These are the compaction. The 8-cell embryo, in case of mouse, undergoes the rearrangement of intracellular junctions, and as a result we can see the smoothening of its surface. And if before compaction the cell borders were easily distinguishable, just after the compaction the surface of embryo becomes smooth. After that, some cells in this embryo are faced to the outer environment, some of them are hidden inside. So now we can distinguish 2 groups of cells. There is the outer cell mass and the inner cell mass. The inner cell mass will further give rise to the embryo itself, while the outer cell mass gives rise to the trophoblast, the fetal envelopes. So now cells, they restrict their potential. If before the compaction we can see every cell has the potential to give rise to all tissues of the embryo, now they redistribute the functions. Then also you have to know the term morula. Morula looks like mulberry, and it consists of 16 blastomers. Here morula is put on the tip of a needle to show its size, to understand in comparison. Okay, after the morula, the next stage is called blastocyst. Blastocyst, it means because it has a cavity. So the key difference between morula and blastocyst is the cavity which is inside. Also, you can distinguish here the inner cell mass and the outer cell mass. As I told you, this is going to give rise to the embryo. while the outer cell mass gives rise to the envelopes. What is important to mention in this slide is that inner cell mass, if it is obtained out of the embryo, is called embryonic stem cells. And if you hear about embryonic stem cells, you have to understand that this is the embryo itself on the very early stage of its development. There are lots of experiments showing that embryonic stem cells could be retransformed into all the cells of our body. And they are very interesting and promising source for regenerative medicine but they are actively dividing cells. So if they are transplanted into adult organism, they always form teratoma. These are the tumors containing the mixture of different tissues. So themselves, they can be transplanted. They should be retransformed into some tissues, and only after that those tissues could be transplanted into the recipient's organism. But first of all, what do you have to know in this field? These are the three terms you have to distinguish: pluripotent, totipotent, and multipotent cells. If we are talking about the morula, all cells in the morula are called totipotent because they are equal. And if we take only one cell, it can give rise to a new embryo, the whole embryo. Then, if we are talking about the blastocyst and its inner cell mass, then we call the inner cell mass or embryonic stem cells pluripotent. Means they can give rise to all embryonic tissues except envelopes. So trophoblast gives rise to envelopes, inner cell mass only to the embryo. And if we are talking about the stem cells in the adult organism, for example, in the red bone marrow, there are hematopoietic stem cells, the most well known. They are multipotent because they can give rise to the cells of our blood, but not neurons, for example. So they have restricted potential. They still can produce more than one type of cell, but not all of them. So if we are talking about totipotent, pluripotent, and multipotent, we imply the difference in their differentiation potential. The most wide potential in totipotent cells, more restricted in pluripotent, and the most restricted in multipotent cells. Okay, but embryonic stem cells couldn't be widely used due to ethical reasons, of course, because to use them we need to kill the embryo, and because they form teratoma, they form tumors, and also because they genetically differ from patients. So if you want to treat one patient, we need to use the embryo with another genetic profile. And to use genetically identical embryonic stem cells, we need to clone the patient, to create the blastocyst out of his somatic cells or her, and it is moreover is prohibited and technically very difficult. Okay, so about stem cells, we'll continue our conversation next time when we have the topic hematopoiesis. Now another slide showing us the possibilities of the in vitro fertilization technologies. So as we have already discussed, those technologies, they could help couples with different problems, male-female infertility, but also some couples, they undergo these procedures not because they have problems with fertilization. Maybe they are young enough and they can have their children without problem, but they are afraid of genetic pathology. For example, if they have high probability of giving birth to a child with genetic defect. For example, if they both have the same defect in their families. To avoid that, they create an embryo in vitro and using the methods of in vitro fertilization. And after that, they use so-called preimplantation genetic diagnostics. It helps them to analyze the cells before the implantation of the embryo. How it could be performed? First of all, one of those blastomers could be obtained, as it is shown in this case. So micropipette is used to aspirate on the one cell. Only one cell is enough to assess the number of chromosomes in it, to check whether exact mutations are present there. And after that, remaining cells could be implanted. But this procedure is rather harmful, and it decreases the probability of implantation and embryo survival. So the success of the in vitro fertilization is lower. So another method is used when cells are obtained on a later stage of development before the implantation. This is what we call the Blastocyst biopsy. As you can see now, cells are divided into trophoblast and the embryoblast. And exactly trophoblast cells are used for the diagnostics. And in this case, several cells could be obtained, and it is also harmful, but less harmful than obtaining of the blastomer. So what happens then with those cells? They undergo either fluorescent in situ hybridization. This method we have discussed last time. It uses specific DNA probes, fluorescently labeled, and it helps to reveal different chromosomal abnormalities like deletions, translocations, and ploidy. So in case chromosomal abnormalities are suspected, then this method is used. Another method is microarrays. These are the chips or small solid fragments of solid material with many DNA sequences fixed in it. Each DNA sequence contains a mutation, exact mutation widely spread in our population, and the DNA of those embryonic cells is hybridized with this microarray. As a result, if hybridization occurs, it means that this embryo has one of those mutations. And this method helps to detect the point mutations, different polymorphism, and others. Also, exome sequencing could be performed when DNA is sequenced, only those DNA fragments responsible for protein synthesis. As last lecture we have discussed, there is only less than 1% of DNA encoding proteins. So this procedure is more expensive, time consuming, so it is not so widely used. But in any way, you have to be aware about these possibilities. And really, if a couple has high probability of genetic mutations, this is the case how they can avoid them. Then, after the process of cleavage, embryo now arrives to the uterus, and it is almost ready for implantation. But to perform this process or reaction, hatching should take place. So hatching, what is it? Like a chick hatches from the egg shell. This in the same way, hatching is performed by the human embryo. Inside the uterus, it gets rid of its zona pellucida or fertilization membrane, and now it comes in contact with the endometrium. The process is very important because this membrane protects from preliminary implantation, and if it doesn't occur, implantation is impossible. Again, several contraceptive drugs, they make implantation impossible because they prevent the production of mucus by the endometrium, so embryo cannot attach to it, cannot invade them. So the implantation is a critical period, and to realize the implantation, hatching should be performed. And the last reaction, this is the implantation. Implantation is when embryo comes in contact with the endometrium, and when it attaches to the endometrium, we call this phase adhesion. When it starts to digest the uterine endometrium, invading inside it, we call this invasion. So implantation, it has like two steps. For adhesion, the presence of mucus is very important. So usually implantation occurs close to uterine glands, producing the mucus in the exact phase of the menstrual cycle. Okay, for this lecture we complete with this slide. Next time we're going to talk about gastrulation and about the formation of the 3 germ layers, their derivatives, extraembryonic organs, and many others. So today, thank you for the attention and goodbye.